Welcome to This Explainer. Today, we're decoding one of the most high-stakes manufacturing operations in existence. It's a microscopic biological supply chain that is happening inside your cells right this very second. We're talking about protein synthesis.
Okay, let's take a quick look at our roadmap. We'll start by meeting the molecular workers, then we'll walk step by step through transcription, where the blueprint is copied, and translation, where the chain is assembled. After that, we'll see how these chains fold into 3D shapes, what happens when the factory actually breaks down, and finally, how modern science is hacking this entire process.
All right, Section 1. Meet the molecular machines. Let's introduce the overarching heroes of our story. These are the microscopic workers that literally do everything in our bodies.
You know, proteins really aren't just generic macros on a nutrition label. They are the ultimate biological multitaskers. Think about it. If you need an enzyme to digest your lunch, well, that's a protein. Need structural integrity? That's collagen acting like microscopic scaffolding. They are our transport trucks moving oxygen, the frontline antibodies of our immune defense, our hormonal messengers, and the literal motors that allow our muscles to move. It's the whole shebang. Now, the crucial mechanism behind all of this is what's known as the central dogma of molecular biology. DNA to RNA to protein. It's a strict rule. You absolutely cannot skip a step. Information has to flow securely from the permanent master storage in your DNA over to a temporary messenger RNA, and finally into the functional protein itself. It is the ultimate rule of biological manufacturing.
Moving right along to Section 2, Transcription, copying the blueprint. Let's head over to our first major phase on the factory floor, where we have to safely access that master code locked deep inside the DNA vault.
So in eukaryotes like us, this copying process happens very safely inside the nucleus. The famous DNA double helix literally unzips, which allows just one single strand to act as our template. Then, our molecular copying machine, an enzyme called RNA polymerase, latches right on. As it reads the template, it produces a complementary messenger RNA molecule. Boom, we've just created a temporary, highly mobile work order.
But before we send this work order out, let's meet the key RNA personnel running the assembly line. First up, we have our messenger RNA, carrying those vital instructions. Then we have transfer RNA, and you can kind of think of these as specialized delivery trucks, bringing specific amino acids to the site, using an anticodon to match the mRNA. And finally, ribosomal RNA, which physically makes up the factory machinery that actually forges the bonds between those amino acids.
Now, you might be wondering how the instructions are read. Well, the genetic code is read in precise blocks of 3 nucleotides, called codons. So if the DNA sequence reads TACGAACCG, transcription translates that into an mRNA sequence of AUGCUUGGC. That precise sequence acts exactly like a barcode, telling the factory exactly which raw materials to pull next.
Let's jump into Section 3, Translation, assembling the chain. With our RNA work order in hand, it's time to hit the fast-paced assembly line out in the cellular cytoplasm. Out in the cytoplasm, the ribosome machinery is just frantic. It operates in 3 distinct stages. It starts with initiation, where the ribosome locks onto the mRNA, hunting for the start codon, which is AUG. Then, elongation kicks in. Those tRNA trucks rapidly drop off matching amino acids, forging peptide bonds to build a growing chain. Finally, termination. The moment the machine hits a designated stop codon, the assembly line instantly halts and releases the newly minted protein.
Which brings us to Section 4, folding, the final 3D shape. Because the assembly line might be finished, but a linear string of chemicals is totally useless until it takes on its final form. This geometric snapping into a 3D shape is what turns a generic chain into a highly specialized tool. And this fold dictates absolutely everything. I mean, the protein's function, its stability, its biological activity, the whole works. And it isn't magic. It's incredibly complex physics, driven by the exact sequence of amino acids, their chemical repulsions and attractions, and the surrounding cellular environment.
Let's do a quick comparison here. If you compare simple prokaryotic bacteria to a complex eukaryote like a human, the factory floor plan actually looks quite different. Eukaryotes have a highly secure nucleus for transcription and require extensive RNA processing, like splicing, before anything gets built. Prokaryotes, on the other hand, lacking a nucleus, do everything simultaneously right out in the cytoplasm, and they even use slightly smaller 70S ribosomes compared to our larger 80S machines.
All right, let's move to Section 5, when the factory breaks. We need to slow down a bit here and zoom out to the macroscopic human level to look at the darker side of this process.
It's a stark question, isn't it? What exactly happens to a human body when the cellular factory breaks down or misreads the instructions? Because, as we've just seen, this sequence has to be unbelievably precise.
Sadly, tiny microscopic typos have massive, devastating consequences. A single genetic mutation in the blueprint builds an abnormal protein, directly causing diseases like sickle cell or cystic fibrosis. But even if the blueprint is absolutely perfect, if the physical folding process fails, those misfolded proteins clump together, and they are the primary culprits behind Alzheimer's and Parkinson's. And what if the factory's off switch breaks, leading to unchecked protein production? Well, that is a hallmark of cancer. Let's look at section 6, hacking the cellular factory. Because here is the incredible news. Humanity isn't just standing by. We have figured out how to actively hack this very biological supply chain.
By mastering transcription and translation, we really unlocked modern biotechnology. We now engineer cells via recombinant protein production to literally churn out human insulin. We're using gene therapy to actually overwrite defective blueprints to restore normal production. We can synthetically stimulate immune responses for vaccines and engineer cells to build entirely new, bespoke proteins.
And now we are combining biology with artificial intelligence. For decades, predicting exactly how a one-dimensional string of amino acids would snap into a three-dimensional structure was an impossibly complex math problem. But today, AI tools can predict these folding structures with astonishing accuracy, unlocking the specific functions of millions of unknown proteins. It's a real game changer. Armed with that AI insight, scientists are no longer just observers anymore. They are basically molecular architects. Through protein engineering, we are tweaking existing proteins to design ultra-efficient enzymes, entirely new classes of targeted medicines, and even revolutionary biodegradable industrial products. And of course, the ultimate hack of the central dogma, mRNA technology. Instead of manufacturing a protein in a massive lab, we can synthetically print the mRNA work order itself. By delivering this synthetic mRNA right into your body, we temporarily turn your own cellular factories into on-demand drug and vaccine manufacturers. No way, right? It's just brilliant.
As our source material elegantly points out, understanding this process forms the absolute foundation of genetics, molecular biology, and biotechnology. Protein synthesis isn't just textbook trivia. It is the fundamental mechanism that converts abstract genetic code into functional, breathing life. So I want to leave you with this final thought. We started by merely observing the biological blueprint, but today we hold the keys to the assembly line ourselves. With synthetic biology and AI right at our fingertips, what new molecular machines will humanity choose to build next? Thanks for exploring the cellular factory with me today.